Well, hey, AP Bio students, are you ready to review? We're going to start reviewing for our AP test that's coming up here. So, what I want to go over with you are just some of the major concepts from Unit 1, our Chemistry of Life. That was actually chapters 1, 2, and 3 from our textbook. So with that, what I am doing for all of these unit reviews is I'm actually just using directly what College Board has provided me as an instructor from the course exam description. You do have this PDF. This is linked on Schoology for you if you want to read more details than what I even provide you. But for each unit, it gives me an outline of what we should be talking about. After each unit, what I would like for you guys to do, I would like for you guys to review chapters 1, 2, and 3 from your textbook, review your test results from chapters 1, 2, and 3, review your Unit 1 progress on AP Classroom. We did the multiple choice progress, so I would do that. And then once you're done with that, I have opened up the Unit 1 progress check for the FRQs. So give yourself time to do the 2 FRQs that College Board has prescribed for you as well to help you with Unit 1. So with that, let's go ahead and talk about some of the main concepts from Unit 1. This was your intro to AP Bio. And with that, it's kind of your shell shock of we're not in regular biology anymore. This is a lot different than what we are expecting. So with this, it's the chemistry piece. So of course, we started out with just basic chemistry content and understanding that. But here's our overview of the topic. So here's the unit at a glance. We talked about the structure of water and hydrogen bonds. We talked about the different elements of life. And then macromolecules, 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 which you guys all really, really loved so much. So we definitely need to do that. This is only about 10% of the exam. But as you guys know, macromolecules are embedded into a lot of different themes, especially when we talk about protein synthesis, when we talk about any genetic change, we talk about DNA replication. And so all of that is an important factor. So let's go ahead and start with topic number 1. So with topic number 1, it's structure of water and hydrogen bonding is what we need to be able to understand. With that, the enduring understanding from this unit is that living systems are organized in a hierarchy of structural levels that interact. So in this topic, we are talking about the atom level, so at that atomic level, to the molecule level, and then also the macromolecule level, but understanding how things like atoms and molecules and slight changes in those can affect the entire system. When we actually got to ecology, we understand that even lacking of certain atoms in the soil changes the entire productivity as well. If soil is lacking in nitrogen or it's lacking in phosphorus, then the plants don't have the building blocks to make the macromolecules that are necessary. Now, in this particular topic, we are talking specifically about water, so we need to make sure we understand the properties of water that result from its polarity and hydrogen bonding effects on its biological function. So with this too, there's some terms that are down here when you look at the slide, talking about hydrogen bonds and in water that results in cohesion, adhesion, and also surface tension. So those are some terms that we should make sure that we are understanding. So this particular diagram should make a lot of sense to you. When you look, here's our basic water molecule, looks like Mickey Mouse. So here's our oxygen, here are the 2 hydrogens. Remember that with this, oxygen is more electronegative, so there is a polarity. So this hydrogen end is slightly positive, and so the oxygen end is slightly negative. So this is a polar molecule because of this uneven distribution of electrons about the more electronegative oxygen. And therefore, this hydrogen, that's just this one proton now, because oxygen's hogging its electron, these hydrogen protons are actually now attracted to more electronegative atoms of other molecules. So here I've got my molecule of water, but this hydrogen of this water is attracted to the oxygen of a different molecule. That's a hydrogen bond. So make sure you understand that this is that hydrogen bond. It's not the bond between this oxygen and this hydrogen. That's a normal covalent bond. The hydrogen bond is a very important bond in biology. Now, when we take a look at this particular oxygen, this diagram is slightly incorrect because there actually should be another oxygen, another water molecule that is actually bonded here. Every water can actually make 4 hydrogen bonds with other water molecules, and that gives water a lot of its different properties. We know that water is a universal solvent. So here's just an example of water being the universal solvent of any charged particle. So anything that's polar, anything that's charged, water can dissolve. So with this, we can also see with this hydration shell, here's a negative ion. So this would be the chloride ion. But with that, now you're actually seeing water surrounding this with the hydrogen side of water surrounding the negative. Here I have my sodium, my positively charged sodium. And so now I've got my water oriented where the negative oxygen side is actually now facing that positive ion. But as far as solubility, that's an important feature of water in all living things, is just being that universal solvent. Remember, some of the other properties of water is that water has a very high heat of vaporization. It also has a very high specific heat, so it takes a lot of heat in order to get it to change temperature one way or another. And that high heat of vaporization also for sweating and cooling, we'll talk about that in a second. But water, with their hydrogen bonds, the properties of cohesion and adhesion, we need to make sure that we're remembering that. When we say that water is cohesive, water likes to stick to other water molecules. When we say that water has adhesive properties, water likes to hydrogen bond with other molecules. So with this too, because of this high cohesion and even adhesion here on water, this is why there's actually a lot of organisms that can actually walk on water, because water being stuck to other water molecules creates this surface tension that's at the surface at this change of where water now meets gas or meets another surface. So here we have this water strider who is walking on water because of the surface tension. This is also why it really hurts when you do a belly flop in water, because you are not, you are trying to break a lot of hydrogen bonds on that water. Now, some of the other properties that I did mention when we talk about water, adhesion, cohesion, um, transpiration is a huge, huge process as far as the water cycle is concerned. And so water also, this is actually showing you water potential in this change. So we'll deal with water potential more in Unit 2 review is what we will address. But this movement of water, even through these small little xylem, water is adhering to the walls of these dead cells, and then water is also adhering to water. So we actually have this cohesion and adhesion that's actually happening within the xylem to help with transpiration. Now, in this particular picture too, we sweat. The reason why we sweat is because water has a high heat of vaporization. And so by putting water on the surface of our skin, for it to now turn from liquid to gas, that actually removes a tremendous amount of heat from your body to now transfer to the water, so it can now phase change. We also know that water is actually the most dense at 4 degrees Celsius. So this is why as water cools in aquatic situations, well, water is going to cool and cool and cool and drop down, but at 4 degrees Celsius, when it now hits its most dense, water then when it freezes, the density expands. And so this is why we have aquatic environments that end up having these frozen insulated areas. And so then there's still aquatic life that can live underneath those frozen areas. And water too, this is why our whole ecosystem and our earth is very, very stable temperature wise, because of that major property of water, that it does have a very high specific heat. And so it's responsible for a lot of our currents. It's responsible for our moderate temperature that we have here in California because of the cooler water absorbing some of that heat that's moving by. So just be aware and make sure you're reading about just different properties of water and why it's so important to living things on multiple levels. Now, when we address topic two is, I'll move myself up here, is really understanding here just the elements of life. When we talk about this complex organization of living systems, requires this input of energy and the exchange of macromolecules. So we need to be able to describe composition of macromolecules required by living organisms. So especially here, we are talking about carbon, we're talking about nitrogen, we're talking about phosphorus, we're talking about oxygen. So it's making sure that we are really clear about the basic elements of life. So make sure you guys are reviewing your carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These would be some of the basic elements of life that we need to be aware of in terms of macromolecules. Remember, all macromolecules are going to be having carbon and oxygen in them, and hydrogen. But then with that, nitrogen, phosphorus, and sulfur do differ in our macromolecules. So we need to make sure that we are paying attention to how those differ. So this is where in lipids, remember that when we're talking about lipids, primarily carbon and hydrogen. So these are long, long chains of hydrogen and carbon. We'll talk then about your monomers and polymers, though, that actually do have some oxygen with them. Your carbohydrates are going to have carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio as a monomer. But your DNA and RNA, now we're going to have nitrogen and phosphorus in DNA and RNA, and proteins are going to have nitrogen and sulfur, because remember that sulfhydryl group that is in some of the R groups of some of your amino acids. So do remember, this is also where in labs, if we were trying to determine what was the genetic material or the transforming agent, we're trying to determine DNA versus nucleic acids versus protein. This is why we use radioactive phosphorus to tag our nucleic acids, and we could use radioactive sulfur to tag our proteins. But we would not be using nitrogen because that's not going to differentiate between your nucleic acids and your proteins. And we're definitely not going to radioactively tag carbon or hydrogen or oxygen because that's not going to differentiate any macromolecule at that point in time. So make sure you understand just some of the basic elements and what macromolecules they would be a part of. Now, in topic 1.3, 1.4, and 1.5, it really is all about your macromolecules. In 1.3, this introduction is making sure that you are understanding monomers and polymers, your monomers, the polymers, and then really it's this bonding that is true for all 4 of our macromolecules, your hydrolysis and your dehydration synthesis reactions. So when we do talk about our dehydration synthesis reactions, this is the joining of monomers in order to form one large polymer by removing water. So remember that this is an anabolic activity. So being an anabolic activity, energy is also required too when we are now putting large molecules together. Now, if we are now breaking those molecules apart, we are talking about hydrolysis, and in hydrolysis reactions, when we are trying to break those bonds between 2 monomers, we have to actually put water back into it, break the bond, but breaking the bond releases energy, so those are catabolic reactions, but we have to satisfy bonding rules, so that's why we're actually using the water in order to satisfy bonding rules when we have now broken that covalent bond. But do remember that anytime we are doing dehydration synthesis, we are building polymers. Anytime we are performing hydrolysis, we are breaking down polymers into smaller subunits. So if I refer to hydrolytic enzymes that are in lysosomes, hydrolytic enzymes are breaking down macromolecules. Now, when we do address 1.4, properties of biological macromolecules, take some time to go through the details in here, in 1.4 and 1.5 specifically, because when we're talking about properties and then we're also talking about structure and function, that's what 1.4 and 1.5 is. There's some very specific terminology in here about nucleic acids, about proteins, about carbohydrates, and about lipids that you really need to make sure that you are paying attention to. I may not hit all of them as I review, but I'm expecting that you guys are going to go back through and you're going to read through these and understand these key terms that are addressed. Now, when we do address macromolecules, so with macromolecules, you should recognize if you see pictures like this, go, oh, that's a carbohydrate. And if I see a picture like this, I'm recognizing it's my fatty acid. Or I'm looking at, if I'm talking fatty acids, I know I'm addressing lipids. If I'm talking amino acids, I know that's the monomer of proteins. If I'm talking about peptide bonds, I know that those are my bonds between amino acids. Nucleic acids, the basic monomer is a nucleotide. So we'll talk more about those, but you should know your 4 major types of macromolecules as far as recognizing their structure, understanding their general function, and understanding how changes in that structure can then change the function too. Now, when we do talk about function, so this was 1.5, and I said go back through. You've got the PowerPoint in order to look through. So you can actually just slowly go through and read these pages, or you can look at the College Board summary as well. Look at these key terms. But let's go over some of these pictures and diagrams about these different, our different Macromolecules. So we're recognizing here let's talk carbohydrates first. So we know that a monosaccharide, that's a monomer. A polymer is going to be starch or cellulose or glycogen would be some polymers you should be aware of. And then why they have a picture here of a chloroplast is remembering that your chloroplasts are responsible for photosynthesis, and this is where our macromolecules all are formed. Now, when we talk nucleic acids, you guys should be remembering nucleotides. Your nucleotides have three parts to them. There's a phosphate, a pentose sugar, and there's four different nitrogenous bases in DNA, and then there's a different one in RNA. So there's U instead of T that we'll address. Understanding DNA, and then also understanding that your nucleic acids are going to be involved in coding information for making proteins and also in directly building proteins. Now, when we do talk about proteins, here's your basic amino acid, written slightly different. Remember, all amino acids have a carbon core. They have a hydrogen attached to them. There's a carboxyl group. There's an amino group. And it's always a carboxyl group that bonds to the amino group of the next amino acid. And then the interchangeable part of an amino acid is this R group or side chain. Remember, there are 20 different R groups for amino acids, and those all have different properties to them. So changes in amino acids can now give changes to the overall protein structure. So when we do talk about polymers of proteins, we're talking polypeptides. And so your polypeptides are everything. You are different from somebody else because of your protein composition. Now, lipids, what we have to remember with lipids is the basic monomer of a lipid is a fatty acid. Fatty acids are long hydrocarbon chains with a carboxyl group at the end. So this is where the oxygen comes in. And then most polymers that we address are going to be your triglycerides. They're going to have one glycerol and three fatty acids. But then also the most important lipid we need to know for AP Bio is we have to know our phospholipids for all your membrane structures. So what I put on the PowerPoint for you is just a review of DNA versus RNA as far as the differences in the pentose sugar, the difference in the nitrogenous base, remembering that DNA is double helix, RNA is just a single strand. Make sure you're remembering where the covalent bonds are, and then remember where the hydrogen bonds are as well. When we talk about lipids, make sure that you can recognize your three different types of lipids. We've got our triglycerides. A phospholipid then is going to be different from a triglyceride in the fact that there's two fatty acids, and then there's this phosphate group that gives it a charge. So this is why this is an amphipathic molecule that is going to actually dissolve in water here and then actually turn away from water, be hydrophobic in this area. Because the important property of lipids that we need to be aware of is that lipids are nonpolar. So since lipids are nonpolar, they do not dissolve in water. And so with that, that's going to allow for a lot of different properties from that cell membrane because of that lipid bilayer that phospholipids form. Also, when we have different steroids, remember steroids, we need to know them in terms of cholesterol is one that you absolutely need to know about. Steroids are lipids, meaning again that they are not soluble in water, but this also means that steroids, some of these structural lipids, and also then some of these hormones, these chemical messengers, they can go right through the lipid bilayer of any membrane because they are lipid soluble. Now, when we talked about cholesterol, we'll talk about that in our Unit 2 review, but remember cholesterol gets used in the cell membrane in order to make sure that it doesn't solidify. These fatty acids here, we need to recognize that these fatty acids that are pictured here are saturated fatty acids. And in animal cells, our cell membranes are made of saturated fatty acids, and what we know is that saturated fats at room temperature are solid, which we need fluidity in the cell membrane. So that's where, again, we talked about cholesterol being embedded in the lipid bilayer of animals in order to make sure that we maintain that fluidity. Now, remember that plants will actually now create double bonds. And so there's more unsaturated fatty acids when there's one or more double bonds in that fatty acid chain. So that's just kind of a review. Make sure you understand saturated versus unsaturated fatty acids as well. Now with proteins Proteins, absolutely vital that you remember with proteins. You remember the difference between the primary, secondary, tertiary, and quaternary structures of proteins. When we are talking about the primary structure, so here I've got my amino group that was just hanging out here. It's not bonded to a carboxyl group. So this would be the first amino acid that would be laid down in this primary structure. This would be your met, um, which would be that start. But then we'd have all of our amino acids, and then this ends at a carboxyl group. This carboxyl group would be the C terminus. So when you guys read about what that is, so this would be the end of the polypeptide chain that is being formed. But do remember then that that primary structure is just the beginning. This does not have function yet. So the second level, this is all the hydrogen bond interactions, are going to either cause an alpha helix or a beta pleated sheet type of shape to it. So that's the secondary level, but that's all hydrogen bond interactions that are going to form that secondary structure. Primary structure was established based on the genetic code. Secondary structure is determined based on hydrogen bond interactions. And then R chain, the R group interactions are going to now start forming this tertiary structure. Remember those 20 different R groups on amino acids, they all have different properties. Some are polar, some are nonpolar, some want to covalently bond, some want to ionically bond. And so they all have different behavioral differences. Now remember that all proteins have a tertiary structure, but some proteins have a quaternary structure where it's two or more tertiary structures that now group together to function as one big protein. Remember your hemoglobin molecule. Hemoglobin is a quaternary structure. So with that, we need to make sure that it was two different tertiary structures that were now forming that ultimate quaternary structure. What we'll also address is when we get to enzymes, remember all enzymes are proteins and you are proteins as well. Remember some of those things that can denature proteins, changing their shape. When we change the shape of a protein, it also changes its function. So pH, temperature, those are things that can actually change that protein structure, some environmental factors that can change it. Remember with your carbohydrates, this is a carbohydrate that is in an aquatic solution. So here's your glucose, your monosaccharide. It forms this ring shape. Remember that in organic chemistry, we actually help to clean it up by not showing you all the carbons and hydrogens that would be at every joint here. But this is your glucose, your C6H12O6 that is pictured here. But you've got this final bond of oxygen here. Now remember that glucose molecules can now form bonds, and so through dehydration synthesis reactions. And so with this, here's a sucrose molecule that's a disaccharide. Here's a polysaccharide. But when we start talking about some of these disaccharides and these polysaccharides, what we have to recognize is there are different ways that these glucose molecules can bond, and those different arrangements give different properties. And that's the most important thing when we talk about some of these polysaccharides. We know that cellulose, that structural component of all cell walls, that's not digestible by most living things. There's just some bacteria that can actually have that actually has enzymes that can break this type of bond. Because what's happening in this type of bond, remember here with this type of linkage, I've got a glucose facing upright here. This one is upside down. This one's upright, this one's upside down. And based on this bond, well, again, we as humans, no animals have enzymes that can actually break that specific bond and perform a hydrolysis reaction on that. Now, if we're talking about glycogen, glycogen is a storage molecule for animals. So this glycogen polysaccharide, it's got different bonds to it, and it also starts forming this branching pattern. So anytime you guys go to Dutch Bros and you get your 100 grams of sugar in your Rebel, well, that excess sugar has to be saved for potential later use. So your liver is going to bind up all of those extra glucose molecules and turn it into this glycogen, and hopefully use those reserves later by breaking off these branches. Now, in plants actually create starches, and starches are then again another polysaccharide, but it's a plant polysaccharide that's for energy storage as well. But again, if you start looking at the bonding is different. You are not held accountable to know exactly the name of the bond, but know that how you attach these glucose molecules now gives it different properties. And so be aware of starch, glycogen, cellulose in terms of their general functions, but also then in general what makes one different than the other. Now, in topic 1.6, you definitely need to make sure that you are understanding nucleic acids in more detail. And so with your nucleic acids, it's knowing the structural similarities and differences between DNA and RNA, especially making sure that we are understanding 5' versus 3' ends. So with that, if we're taking a look at our basic DNA, we should know that your DNA nucleotide is going to have deoxyribose. And so down here, we can actually see in this hydroxyl group, there's not an oxygen here. There's where in your ribose sugar, there is this hydroxyl. Here, there is no hydroxyl, it's just hydrogen. Therefore, deoxyribose is this pentose sugar. This is ribose. Now, this makes this more reactive, so this doesn't last as long as DNA. This is a much more stable molecule. This is my nitrogenous base, and then I've got my phosphate. So this is my nucleotide of 3 parts. In DNA, this nitrogenous base, remember purines and pyrimidines, I've got GCAT, and remember the mnemonic I taught you of GCAT, puppy, puppy. So with GCAT, puppy, puppy, G is a purine, C is a pyrimidine, A is a purine, T is a pyrimidine. Remember that G and C always connect together in the middle of this double helix by hydrogen bonds. And thymine and adenine always bond together. Those are your complementary base pairs. And remember, it's hydrogen bonds that actually connect these. Now, the hydrogen bonds, remember I taught you GCAT puppy puppy 32 because guanine and cytosine want to make 3 hydrogen bonds with each other. Adenine and thymine want to make 2 hydrogen bonds with each other. Then also remember that when polynucleotides connect to another polynucleotide, they have to be anti-parallel. So remember that term in terms of the shape of DNA, or even if we're now talking about transcription, the polynucleotide, one nucleotide, if it's hydrogen bonding with the other side, one has to be upside down compared to the other. So remember that there's directionality to our polynucleotides. 5' end is where now my phosphate is the end. If I have a hydroxyl, that's my 3' end. And those are in reference to which carbon on the pentose sugar it's bonded to, but it's easier to just remember phosphate 5. If I've got a phosphate sticking out, the fuf and fuf, phosphate is my 5' end. Oh, I don't have a phosphate here, so that's got to be my 3' end. RNA also has a 5' end and a 3' end as well. Again, phosphate, 5' end. The other difference between RNA is going to be single-stranded in animals. Remember, we talked about viruses, though, can be double-stranded RNA. But when we do talk about our RNAs, remember we've got uracils instead of thymines. And so now I'm going to have GCow is what I'm going to have. I'm going to have G and C. A and U as my bonding pairs is what I'm going to have with those. So in section number, in unit 1, let's just remember what we need to be reviewing. This truly is an opportunity for you guys to just review and go back through those particular chapters, chapters 1, 2, and 3, pay attention to macromolecules. But after you watch this, after you've gone through those 3 chapters, make sure you then go onto the College Board website, to My Classroom, and do the Unit 1 progress check is what you guys should be doing. Take some time to practice those FRQs, since this year's 2020 test is just going to be 2 FRQs, one long, one short. So practice FRQs. So with that, I will talk to you guys later, and hopefully this helped just kind of summarize some major concepts from Unit 1 for you. So with that, I will see you on Unit 2.